That's cool. Good morning, everyone, and thank you for attending the first bioarchaeological lecture on Introduction to Archaeobotany. To shortly present ourselves, I'm Caroline Douché, and my co-lecturer is Alexia Decaix. We are both postdoc researcher in archaeobotany, leading project in the Caucasus for Alexia and in Iraq for myself. So archaeobotany is, Alexia, can you just switch? Yeah. Archaeobotany is dealing with plants, which can be described as living organism, usually composed by root, or also called shoots, stem, leaves, flowers, fruit, and seeds. Plant can be divided into the vascular and the non-vascular plant, the distinction being the presence of vascular tissue called xylem and phloem to transport water, nutrient, and food. Among the vascular category, we can find plants that produce seeds, the gymnosperm and angiosperm that you can see on the right bottom of the screen. Gymnosperm produce plant in cones. The seeds are naked, meaning they are enclosed in an ovary. They are not enclosed in an ovary. In this category, we can find the ginkgos and the conifers, like the pine tree you can see at the top of the screen. In contrast, angiosperm produce seed from the flowers with seed enclosed in a closed ovary. Among the angiosperm, we distinguish the monocotyledon, which have a plantlet with only one cotyledon, like cereal. Here you have an example of barley. And the dicotyledon, which have two cotyledon then. And among this category, you can find the pulses with here a photo of a chickpea. In order to understand the interest of studying plant in ancient societies, let's have a look to the wide range of plants used in daily life. So first of all, some plants are edible and will be used for food or drink for human and animal consumption. That's, for example, the case of barley. That can be transformed or processed to make bread, soup, beer, and given raw to animal as fodder. Other plant or plant part can be used for medicine, in infusion, as a smoke or gel can be used, or cosmetic purposes. Plant can also be used for craft activities, fiber for textile like flax or cotton, and a wide varieties of wild plants. And also for dyeing. They can be used to make tools or be added to clay to reinforce pottery. Herbaceous plant and wood can be directly used as fuel or as a starter to make fire. Or indirectly, like dung cake that contain a plant eaten by the animal. Finally, wood and herbaceous plant can be used in the construction, architecture, roofing, and boat. And in addition, plant can be used for their nice smell and colors, like ornamental plant grown in the garden, or as a symbol, like for fertility, for example. So as we can see, plant can be associated to a wide range of use, depending on when we are catching the story, interpretation will vary. In archaeology, we will face two situations: the recovery of plant in primary deposit, meaning they will be found in exactly as they were disposed or used. And this is, for example, the case of granaries. And on the other hand, we will find the secondary deposit where the assemblage is mixed and it can be composed with plants from different origin, different place, different period. And this is, for example, the case of midden or garbage.
So plants are everywhere in our daily life, and in order to reconstruct environments and activities, we will refer and combine several disciplines connected to archaeobotany. Obviously, the name is a clue. We will first refer to archaeology and botany to understand and define what we are looking and where it comes from. But we will also have to include notions of ecology, biology, and biochemistry to understand the ecosystems, ethnography, experiment, and statistics to interpret the results. The thematics covered by research in archaeobotany are very large and diversified, but whatever the region or the period studied, archaeobotany has two main aims: reconstruct past environments using ecological specificities, and explore human activities such as practices related to farming or transformation like cooking or crafting, for example. The two are complementary since they are strongly correlated to each other, but they usually refer to different material, disciplines, and methods. So today, the lecture will be divided in two parts. First, the pollen, and then the study of phytoliths. So let's start with pollen and its related discipline, palynology. Based on the definition given in the Illustrated Pollen Terminology published in 2018, the palynology is the science of palynomorph, including pollen, spores, cysts, and diatoms.
So here we will focus on pollen, which is a male element of sexual reproduction of flowering plant and conifer. To briefly remind you, pollen is formed in the anther of the seed plant. The male organ that you can see in yellow on the left and top right photos, sketch, and is released to reach the female. As you can see on the bottom sketch, representing the anatomy of pollen, they are composed by different layers, but it is the out layers, which is called the exine, which is the most resistant and usually preserved in fossil pollen. So Mesopotamian and Egyptian knew very well the pollen since they were using it to cultivate the very famous date palm. So date palm is dioic, meaning that male and female flowers are located on different trees, even if it's not botanically correct. So to obtain fruit, hand pollination is required. So the farmers takes an inflorescence from the male palm and spread it on the female flowers. So you can see on the pictures that's what they're doing. And on the right picture, you can see the male inflorescence on the flower, on the female flowers. However, even if we know that this very traditional ancestral knowledge exists, due to the micro size of pollen, the development of research in palynology is associated to the development of microscopy. During the 19th century, the first classification based on the morphology of pollen was described and the chemical method of extraction was established. During the 20th century, the importance of stratigraphy appears and the first pollen diagram were published. But palynology was mostly related to ecology, and it also had the economic advantage since it allowed to extract charcoal. And it's only during the 1950s that it developed in other fields of studies, including archaeology.
So soils who support the plants can contain pollen that can be recovered through sampling. Samples for pollen can be collected in natural environments and will therefore reflect natural vegetation and its chronological evolution. But samples can also be taken within archaeological sites. But there, it is important to remember that they can represent surrounding vegetation and anthropogenic activities. And in this last case, it is better to compare the results with those out of the sites as references. For the natural environment sampling, the way is to take a core, a column of sediment, then describe the different layers, take samples at different depths of the core, and, if any, extract the charcoals for C14 datations. The amount of sediment to be used varies according to the specialist, from 30 grams to 1 liter. And for on-site sampling, the samples can be collected following a column in a trench previously cleaned, of course, or directly in well-defined context. And then we will need around 200 grams of sediment. So to illustrate, sampling can then be collected in specific environments where the chance to have pollen preserved in the core is the higher. These include lakes, bogs, and natural sequences. And to answer specific questions, samples can also be collected in situ, directly in the archaeological deposits, like burial or mummies to investigate, for example, plant offerings, or directly in stratigraphic sections. As we said, the first method of extraction was developed in the second half of the 19th century and improved since then. And in order to be analyzed, the sediment samples collected for pollen require some pre-treatments in lab involving different chemical products. And as we already mentioned also, the fossil pollen are normally represented by the out layer of the pollen, the exine. And once the slide is ready to be analyzed under the microscope, the aim is to observe and define the morphology of the exine, its shape, the presence of apertures, the structure, and to compare it with modern pollen or atlases of reference collections.
So once we have identified the taxa through the fossil pollen, so we have the families or genus of the taxa, and once we have counted them, we can create a pollen diagram. To interpret the diagram, it is important to keep in mind that the pollen abundance will depend on its production and dispersal or pollination modes for each taxa. So the wind being more effective than animal, for example. It is also important, time is also important, since the more ancient will be the sample, the more damage may be the pollen. This also depends on the thickness of the exine, which varies according to taxa and on the context sediment. So here you have an example of pollen, like a very simplified pollen diagram, showing, for example, an increase of a decrease of the forest species, and on the other side, the extension of wildgrass. So it can refer to a change in environment. Among the important palynological work done in the Near East are the pollen diagram obtained from different locations. So you can see on the map on the left. These different diagrams highlight change in the vegetation through time. One important change occurred during the transition from the Younger Dryas to the early Holocene, which also correspond to the transition from Natufian to early Neolithic. And at that time, we observe, as here on the example of the lake diagram from the lake Van in Turkey, an increase of oak that you can see with the red arrow in the middle. So we see an increase of oak, and in parallel, a decrease of the Chenopodiaceae family, indicating the expansion of the forest cover. Indicating the expansion of the forest over the giant steppe vegetation as a result of warmer and moister condition. So the expansion of a more humid climate, allowing the expansion of various species, has been interpreted as one of the factors involved in the emergence of agriculture. But change in pollen record can also reflect landscape management, such as cultivation or deforestation, for example. One of the most famous examples of intrasite pollen analysis come from Shanidar cave in northern Iraq. In one of the burial, Shanidar 4, so the cave is shown in the picture on the left. So Shanidar 4 containing a Neanderthal skeleton. So several samples were collected by Arlette Leroy-Gourand in the 1960s. Two of them, like you can see, highlighted with the red square on the sketch on the bottom left. So two of them were very rich in pollen and even included the remain of anther, suggesting the presence of complete flowers in the burial. The high concentration of a limited range of taxa, so approximately 7, compared to the wide diversity of plant growing in the area, led the specialists to suggest that these flowers were selected and deposited with the dead, either as an ornament or for medical purposes. For that reason, the burial wasn't called the flower burial. However, this interpretation was always debated because the pollen could also come from the surrounding landscape or have been brought by animals through burrows. So there is actually new work carried out in this cave, and one of the aim of this new project is to elucidate the question. So, as we could see, the study of pollen covered research on paleo-environment and human activities. But as highlighted with the example of Shanidar, the interpretation of pollen from archaeological sites is often discussed. In most cases, pollen are widely used to reconstruct past vegetation and indirectly give information on climate change, such as moister and warmer temperature at the beginning of the Holocene. But as we saw, human societies also have an impact on the vegetation, and pollen record this transformation or this impact, like cultivation or deforestation. Phytolith, which is a second type of macrobotanical remain that we are going to present, allow to access similar question, but as we're gonna see, a bit differently. So phytoliths are produced by deposition of silica originating from groundwater absorbed by the plant, the living plant. And the silica is deposited in and between the cell of the whole plant. It is thus a mineral component, not an organic. Silicium has no function in the plant metabolism and therefore travel to the epidermis, so to the peripheral tissue, where the plant evaporates. When the leaf falls, the phytoliths are released, and some are preserved, whereas some are dissolved.
A very small proportion of plants produces phytoliths. Plants in temperate environments generally produce relatively few phytoliths, while plants in tropical environments produce many more. And some plants are very productive, as the monocotyledons, such as the Poaceae, which is the family of cereal. We have here an example of rye. Also the Cyperaceae family, which are mainly herbaceous plants growing in wetlands, as the sedges, or also the Arecaceae family, it's the palm or the date palm family. Also in the ferns group, we find a lot of producers of phytoliths, and mainly in the Equisetaceae family, the puzzle grass family. Other plants are very frequent producers. This is the case of many families of dicotyledons, and in particular, of course, of tropical woody taxa, which means tropical trees. And finally, some plants, indeed the vast majority of plants, are not very productive or produce uncharacteristic phytoliths. And also it should be remembered that phytoliths are very resistant to chemical and thermal attack, but also to biological and mechanical degradations. So phytoliths are casts of plant cells or tissues, and they can have different shapes, as you can see here, characteristic of the cells in which they are formed. So they are characteristic of cells and not directly of the taxa. So classification is therefore based on morphotype and not directly on taxonomy, with two basic principles. The first one, the multiplicity, means that each taxa generally produces several forms of phytoliths, several morphotypes. You have here some example of different morphotypes produced on the left by the grass family, and on the right by the palm family. The second principle is the redundancy, which means that many morphotypes are common to several taxa. So here you have, for example, on the left, an elongated cell that you can find in almost every species of Poaceae, so more than 12,000 species. So very few phytoliths characteristic of a species are therefore known, but large groups are recognizable. The grasses produce many more phytoliths than most taxa, and an interesting thing is that the different parts of the plant produce phytoliths with distinct morphologies. Such thus, as shown in this example, the phytoliths produced in the stem will have morphology different from those produced in the spike. And for grasses, one can then identify the different parts of the plant. The spiky and elongated cells are from the stem and the leaf, as you can see on the graph, and they're among the most frequent phytoliths that we can find. The dendriform cells that are from the parts containing the seeds. You have them at the bottom of the picture. And this is very interesting because this can give information about certain archaeological structure. For example, if we found it on a grinding wheel, it indicates, for example, the crushing of grain. And the bulliform cells are also from the leaf. They allow the opening and the closing of the leaves, and they're very interesting because they indicate aridity. So the morphotypes have little taxonomic significance, but in the case of graces, however, it is possible to identify subfamilies. Thus, there are several subfamilies of graces that do not have the same ecological requirements and do not have the same distribution of the globe. The long cells, as we have seen previously, are the same for all subfamilies of Poaceae. But the short cells, as you have some examples here, make it possible to distinguish subfamilies. For example, the trapeziform, but also the conical or polylobed cells are indicators of the festucoidae subfamily. It's the subfamily of rye. The bilobed cells are indicators of the panicoidae, which is the subfamily of millets. And the saddle cells are indicators of the chloridoidae, which is the teff subfamily. And this latter one indicating arid, very dry, and xeric environments. And phytoliths can thus be paleoclimatic indicators.
So since the phytoliths are mineral components, once a plant dies and disappears, the phytolith can either be dissolved or be preserved in the soil or sediment and last for millions of years. In terms of taxonomy, they have the advantage of being resistant to a wide range of soil pH values, around from 3 to 9, and also to various environmental conditions, including wet, dry, and the alternative one, so wet and dry. In addition, they don't need a specific condition to be preserved. So they don't need to be charred, or they don't need to be soaked in water, for example. As an example to illustrate this, here you can see illustration of very old phytoliths from the site of Dmanisi, which is located in Georgia. And they were dated from 1.7 million years ago. So the good preservation of the phytolith indicate, so you have here the different shape, the variabilities of the different phytoliths recovered in the site, and their good preservation indicate a change in environmental condition in the region. It points to an ecological shift toward increasing aridity, contemporaneous with human presence in this region. The study of phytolith is relatively quite recent discipline in archaeobotany. Although the first observations are dated to the mid-19th century, as for Palynology, the first research were strongly focusing on ecological questions, so to reconstruct the environment. And it is mostly around 2000 that the discipline emerged in archaeology with new research questions like the domestication, and also the development of new methodology to help with the identification using, for example, morphometry, statistic, and more new methods to work on it. So to collect the collect of the sample for Phytolith is quite easy, because you basically just need to clean tool and a Ziploc. But it is important to keep in mind that in order to produce a qualitative result, the best is to be able to compare natural and anthropogenic soil. For that reason, phytoliths are often collected within micromorphological thin section. It is also strongly recommended to sample, in addition to the archaeological level, it's also recommended to sample the surface soil of the site, which will allow taphonomic comparison, for example. As for pollen, the study of phytolith required pre-treatment of the sample, so in the lab, to clean and extract the phytolith particles. Similarly to pollen, at the end of all these different steps, a glass slide will be observed under optical microscope, and the elements will be identified using reference material.
So since phytoliths morphologies are supposed to be characteristic of a plant part, as we have seen, the stem, the leaf, or the inflorescence, they are used to reconstruct human activities. The grass family, including the cereals, is the most investigated one. First, because it produces the higher amount of phytoliths, and secondly, because there is a long history of research explored by also other fields of study, like seeds and fruits, and which can be completed with the study of phytoliths. Among some of the earliest work conducted in the Near East is the work carried out by Patricia Anderson. Within the lithic assemblages, she found elements of tribulum with a glossy useware that could potentially be identified through phytoliths. In order to check if shopping cereals with a tribulum actually leave gloss on the lithic pieces, and to identify which cereals were processed by those archaeological tools, she conducted experimental work on tribulum raining. And results show numerous connected epidermis with very characteristic fractures. Samples for phytoliths can be collected on various farming tools, including grinding stones. And here you have the example of Jaffa armor in northern Syria. Phytolith characteristic of barley inflorescences of the spikes were identified, and combined with useware analysis, the study suggests the use of the stone to either de-husk or grind the cereal. Similarly, sediment samples collected on the site can provide information on crop processing, which means the different steps between harvesting and consumption of those cereals. Here on the right, you can see a representation of the main stages of rice processing, resulting in products and byproducts. Each stage leads to the production of different parts of the plants. The main desirable product would be the grain, whereas the stem and the leaf will be discarded. And since they produce different phytoliths morphotype, the composition of the assemblages can help to identify the different cleaning stage of the samples. Another ongoing research that developed during the last years is the domestication process. And here, as an example, we have investigation on phytoliths, which allowed to work on rice domestication. Indeed, statistical analysis incorporating a different shape and size measurement of the double-peaked gloom cell phytolith that you have on the left picture allow to distinguish wild from domesticated rice. And moreover, the bulliform cell that you have on the right from domesticated rice have a larger number of scale-like decoration than the wild rice.
As we mentioned at the beginning of the lecture, plant can be used as food for human and animal consumption. Animal can be fed by two ways. So if you have a look on the left picture, so you'll see that animal can be fed by two ways: fodder, which means that they are fed with plant controlled by the farmers, or by grazing. So you can see on the right, meaning that the plant originating from the wild or less or uncontrolled environment. Dung is a valuable resource because it's cheap and it's widely available. And for that reason, dung cake are very often dried, collected and dried and used as food for eating, cooking, etc. Apparently much more used for cooking than for heating, but that's another question. So consequently, plant remain, so phytolith, macrobotanical remain, so seed or charcoal, and often spherulite will be preserved in the dung, either charred or in the ashes, and this allows us to recover them in the archaeological deposit. So it is also interesting to collect sample in dung layers. So sometimes you can see them in directly on the site, but usually you see them much better in the archaeological trench. Because the taxa that will be recovered in these layers can reflect herding or animal husbandry systems. Experimental work carried in the Gujarat by Carla Lanciotti and Marco Madella indicate that dung from buffaloes provide the higher amount of phytolith. And then the dung from cattle also provide phytolith, but a bit less compared to buffaloes. So the reason can be related to the type of plant which is added to the preparation of the cake. It can also be related to the type of fodder given to the animal, or even the graze land. Similarly, there was ethno-archaeological study carried out around the site of Bestanchur, so it's a Neolithic site in northern Iraq. And this study highlighted the variation of feeding system, as you can see with the three different tops. So depending on which of the origin of the dung, depending on which animal was fed, we will recover different type of plant part, and so different taxa. And at the Ubaid site of Tel Zaydan in Syria, the presence of spherulite in pyrotechnic features that can be considered as kiln, I guess, demonstrates the use of dung as fuel. And also the study of Phytolith mainly identified the taxa of Cyperaceae and grass inflorescence and leaves, as you can see on the table they published. So during the excavation, you may also discovered white silica skeleton, which is usually very easy to recognize. And they sometimes show a pattern that refer to craft activities such as basketry or matting. So here you have an example from Çatalhöyük in Anatolia, where the collection of samples from different baskets recovered in domestic and funerary areas were recovered. And Phytolith analysis indicate that they were made with panicoid grasses, such as agropyron, that you can see, so the Phytolith and the related plant on the picture. And so this plant may have been used to produce basketry. And on the same site, so at Çatalhöyük, they also recovered, they also observed traces of Phytolith on the floor, and they represent traces of mat that were that refer to reed map, which is still widely in use, as you can see on the bottom right picture. So it can be used in various ways on the floor, but also in the architecture.
So to sum up this first introduction in archaeobotany, dealing with the micro remains, we can say that despite their invisibility on the field, this remain can be very helpful to reconstruct past environments and human practices. The field of study is very large and diversified, as we have seen, since it allows us to access questions of farming activities, including cultivation, domestication, storage, and also food processing, like de-husking or grinding, for example. The main limits of the disciplines for archaeology are first deriving from the biology of the plant itself. For example, some do not produce phytoliths and some do not produce much pollen. And another limit is related to the taphonomy, of course, due to the context and are specific to taxa. And for that reason, it is very important to keep in mind that the best practice is to combine methods and disciplines by applying different micro studies, as for example, highlighted by the recent development of studies associating phytoliths and spherulites to characterize dung. But also to combine micro and macro botanical studies, like, for example, to combine pollen and wood charcoal to reconstruct the vegetation cover. And to better understand the interest of interdisciplinary studies, we invite you to join us for the next lecture on macrobotanical remain on next week. And thank you for your attention, and please don't hesitate to ask questions if you have any.
That was much
Very
quicker. I don't know if there is a chat or